my name is Jeff Martin, and I'm here with Limula, Tom and Caroline. So Tom, Caroline, what is Limula doing?
So we've set out to solve the problem of cell therapy manufacturing.
Interesting. What problem in particular? There's plenty of problems to solve.
Yeah, I mean, in particular is how to make these therapies accessible. It's fantastic what we could do in the lab, and then those first clinical studies, and even a first approval already in 2017. Mm-hmm. And it was exactly at that point that my co-founder Jan was introduced to, we effectively have in our hands a possible cure for these leukemia patients.
Right.
But it's the most expensive drugs that have ever been made.
Right.
And so starting with that problem statement, he worked alone in his garage and 3D printed the first version of what is now our solution for cell therapy.
Feeder. Yeah, so what are we looking at here?
So this is the core of our technology, and that's actually a bioreactor and a centrifuge that is combined. And the idea behind it is that the cells never leave that vessel. So that's what Jan came up with to solve the major issue of the CGT manufacturing. And that means that in our device, you can do everything from the beginning to the end in a simple device, very easy to use, lowering massively the cost of producing those cell therapies, and also lowering the need of infrastructures that are not affordable. And this way, we bring more therapies to more patients.
So what is the max capacity of one of these things? How many would you need, like at a full scale GMP scale up? Like how many of these things would be going at once?
So just like those treatments, you need one dose, one patient, one injection. You need one of these. This is for primarily autologous treatments.
Oh, okay. So then is it like one and done, or are they reusable?
No, it's single use.
Yeah.
So you need one per patient, yes.
Interesting. Okay, see, I'm like so unfamiliar with any of these processes. So apparently, I asked you before the interview, this is the first of its kind. It's the first centrifuge and bioreactor combination ever.
That's correct.
Possibly correct. You don't want to overstate
no,
overstate it. I mean, it looks interesting.
So we've come a long way from what was that initial idea and those runs of Jan working alone.
Yeah.
He then teamed up with Luke and brought me in. So we were three classically trained as chemists that sat around the table and tried to evaluate what do we need to validate that this idea of Jan's is actually a viable solution to the manufacturing and scaling problem.
Right.
And so from the three of us, we started the company in 2020, and we grew the team now to 20 people 5 years later. And we're here at the Congress bringing out what is the first iteration of the technology in a format that we can bring to GMP.
See, it looks much more like a cell therapy manufacturing device in the picture than it does.
Yes.
That makes way more sense.
I brought the picture because we've got at the stand the full version. But
so when so now that I see it, it goes horizontally, and then the centrifugation. Do the cells get collected in this lip here? Where does it?
So
this is totally new to me. So it's
exactly. So you can orientate in the two orientations. So basically horizontally, you can incubate your cells and you can rotate the bioreactor if you need. And then as soon as you want to collect your cells, media exchange, you go that way, and then you centrifuge. And yes, you're right. The cells will just pellet at the edge of the rotor. And then the beauty of it is that you can remove any volume you want, and we are really precise in the volume management. And then you can leave your cells or collect, you can wash as much as you want with very, very high efficiency.
Right. So you guys can do the entire process, even like genetic editing.
Exactly.
All the expansion is done right in here.
Yeah, that's
and
correct.
Then centrifugation, resuspension.
Exactly. So basically from the starting material to the final product, everything in the same machine.
Nice. So you mentioned that you're approaching, or you have your first clinical program, or what is the status?
So we've been
programs?
benchmarking with what are clinical procedures, and I mean, with Caroline joining the team, having worked previously in manual mode in GMP, I think we were selling the dream. Maybe you can expand on what
Yeah.
it was to bring that forward.
Exactly. So when I was in process development, I think one of my frustrations was to never have the right tool, because they are very good tools, but you need many of them. You
Yeah.
have still a lot of manual steps where you need an isolator, you need very highly skilled operators.
Yep.
And then from the process dev to the GMP, we were also lacking tools to make the transition. And when I first met Luc and he introduced me to the technology, I was like, wow, actually
That's cool.
someone is really doing it, because there was so much promise. And actually I never saw it as a process dev user. And so now it became a reality. We
Yeah.
really have a tool that is low barrier access, very cheap, where you can start doing all your process optimization. And then it's exactly the same core technology. You bring it to the Lim1, the big device,
Yeah.
overnight, basically no process optimization, only full automation on the Lim1,
Yeah.
and that's it. So I was looking for it. I discussed with those guys, got excited by the technology,
Yeah.
and joined the company
"I'm
to try
in,
to
we're
make a
doing this."
difference. Yeah.
Nice. So again, I'm relatively unfamiliar with this whole territory here, but could this be used as a benchtop thing, like at a hospital? It seems much more simple than the normal,
This is exactly the design requirements and testing that we put in place. So
Okay.
our vision is that localizing the manufacture removes a lot of the complexity in delivering those cell therapies.
100%.
So that is the key input requirement, is to be able to operate in an environment that a hospital can maintain.
Yeah.
So we have different visions of how far we could go, all the way down to a nurse should be able to do the manufacture.
That would be
It'll be
great.
stepwise to get there.
Yeah.
But most of our initial use cases are clinical research centers, where you have a clinician who also has the ability to do research and treat really the patient that's in front of them. And so we're really
Yeah.
excited that a couple of those centers are planning their in-human study with our device as the manufacturing,
Yep.
coming out within the next 18 months.
Oh wow. Okay. So what is like the timeline then from leukapheresis to reinfusion using one of these? Is it similar time frame, or is it
So it's
shortened?
we've just done the exemplar case in the machine. So
So,
Okay.
exactly. So that's the first time we've proved that actually we can do end-to-end, and we've done that with a proof of concept that is a 7 days CAR-T, which we think is relevant for the field. And yes, the results are very good. So in 7 days, you get a clinical dose of CAR-T cells
Right.
with the right performance, biological performance, and everything in the same machine.
Yeah.
So, yep.
Okay. So cost of goods down,
Exactly.
timeline down,
Yeah.
simplicity up, I guess,
Yes.
complexity down.
and we can't take credit for all of the aspects of achieving that, because we also are very reliant but fortunate to collaborate with reagent providers. Looking
Yeah.
at cell selection, for example, we have shown that in the bioreactor with the reagents that we can do that, and then with the viral vector transduction activation, again, in being able to tune the reagents to the procedure.
Yeah.
Important to us is showing the versatility.
Exactly. So you say cell selection. Speak more to that. I'm sure that, again, I'm unfamiliar with how it works normally, but I'd imagine there's some kind of like beads involved or something. How does that work in this closed
So
system?
for the time being, we are working with microbubbles.
Okay.
So based on flotation, basically, and that's what we just showed. We have an application note that we brought with us, and so we show that in less than 2 hours, you can select your T cells
wow.
with very high
Okay.
performance.
Yeah.
So it's based on the academic reagent, but it works very well, and we have other development projects on that, but based on buoyancy. But we are completely reagent agnostic, so any new reagent
Nice.
on the market we could test, and we are also completely interoperable. So if you want to keep your cell selection as it was before, you
Yeah.
can, and then you can interoperate the two machines, basically.
So let's imagine that a company who is already doing GMP manufacturing wants to transition to this. What is that? Is it a pain to do that, or is it relatively simple to kind of migrate to the system?
So I think there's two levels to answer that question. What
Okay.
is the technical?
Okay.
So there we would say is particularly easy, and this is where we wanted to ensure that we are compatible with many different reagents and different procedures.
Sure.
The other part is more complicated, which is about the regulatory acceptance of making a change in your manufacturing.
Sure.
But what's exciting is that the regulators are becoming more and more accommodating of making changes to already approved procedures. And so there was a big announcement just timed with the conference of JP Morgan in San Francisco from the FDA, really working on how to facilitate changing manufacture of an approved process. And we are
so it's good timing then.
Looking at supporting such changes, in particular because we have a full automation approach that can greatly facilitate scaling out the manufacture.
What do you mean? Fully automated, as in like there's no human in the loop at all needed, or what do you mean fully automated?
Very minimal human intervention.
Yeah.
So once you start your process, basically, very minimal.
I guess if you're doing it all in one location, then it becomes a lot more simple to do fully automated.
Yeah, exactly.
There's no robots needed to transferring solutions and stuff.
And the software is very user friendly and made to be used by
a nurse in a hospital.
Exactly. Yes.
That's cool.
So here we're talking about where we end up, which is clinical grade commercial products being manufactured routinely.
Yeah.
The other thing that our technology allows is facilitated in the R&D. When you are doing very small scale, we actually have a version of the technology that you can use in your manual setting, tune your parameters, find conditions that work, and then facilitated move directly to a full automation without a tech transfer of changing the underlying technology. And so this is a second problem that we solve. How do you facilitate going from R&D to the clinic?
Yeah. It's difficult. A lot of red tape and road bumps.
We want to make it easy.
Yes.
Yeah. So is it a different device, or is it just kind of the same thing that you use?
So it's the same bioreactor, but a different device. So the LimGrow is the entry device, so R&D and process dev. It only allows for rotation, but you can fit it in a regular lab. So all you need is an incubator and a biosafety cabinet, and then you can actually optimize all your parameters for a very low cost.
So are you guys trying to work with universities and stuff as well to get them into just a individual labs?
I mean, university when it's a clinical research center. So very often you find the hospital in combination with the university mandate for doing the research.
Got it.
But research for research is interesting,
Yeah.
but we're more interested in accelerating access for patients.
Right.
And this is really where we've got to as an industry, which is when you look at rare disease, you look at some of those leukemia treatments, they're on market, but patients are still not receiving them. And so we really want to solve for that scalability and access.
Yeah, that's been the major through line of all the conversations today was we have solutions that work. We need to get those things to the actual people who need them. And it's a lot harder.
I mean, it's a fantastic position to be in.
It is.
Coming from the scientist's side, it's been reduced to an engineering problem.
Yeah.
And we are a Swiss precision engineering company to solve what is now reduced to an engineering problem.
Yeah.
And we're very proud to be able to show here at the Facilitate Congress in San Diego, the first time the device in public in the US.
Oh, wow.
And from here, it will actually already go to a biotech that we're working with on their new therapy.
Excellent. So can you do, theoretically, could you do anything besides cell therapies in here? Could you do some kind of AAV manufacturing or anything else? Or is it strictly this is for a cell therapy expansion?
I think you could do anything. It needs to be tested, but we had requests for protein production, and there is no limit. But we are focusing on cell therapy, but I think it's applicable to anything.
Right. Well, it was great talking to you. And again, that's pretty innovative.